Really enjoyed the previous presentation and all the other talks. So hopefully I don't put you to sleep showing you sequences here. But this is sort of my attempt to give you sort of an overview of some of the things that's at NCBI. Some of it may be useful to you, hopefully. So let's start here. I'm not going to talk about the several petabyte of sequences that we have. I'm going to focus on one sequence here and talk about how do we make sense of this. So we have a sequence here. It means nothing at this point, right? It's just a series of letters. How do you get meaning out of this? How do you know which organism it came from? How do you know where it was collected from? And how do you know if you can collect it again? So let's see if we can use some of the tools that's at the GenBank and other archives at the NCBI to answer some of these questions. So in the nucleotide archive, you have your raw reads here. Hopefully people can see my pointer. And then there's all these layers of information on top of these raw reads that one can use to understand more about a particular sequence. So we have assembled data, we have annotated data, there's metadata, and there's various tools to get to that. So, so here we have our sequence. I think some of you will be familiar with BLAST, maybe most of you. That's probably the one interface that a lot of people use at the NCBI. So the obvious thing to do would be to go to BLAST with your mystery sequence in hand. And then from BLAST, you can go to seeing what other sequences it's closest to and learn something from them. And then when you look at enough of those, maybe you can go into publications. We have a whole medical, well, not only medical, but mostly medical journals in PubMed and PubMed Central. But you can also go to external places like INAT or various other websites with information that you get from these individual sequence records. So, so here we have BLAST. So this is how you get to BLAST. It's pretty easy. It's on our webpage. It's the first thing you see in the, well, the fourth thing you see in the popular resources. And then there's several flavors of BLAST. I'm going to stick to the vanilla flavor nucleotide BLAST here. So let's go to that. Okay, and then we paste our sequence into the interface here. So there's a lot of things on this page. I'm not really selecting a lot of other options at this point. I'm just using the default option, but there's video, there's a video here about making custom databases. There's various question marks you can click on if you want to understand particulars about how to do particular things like aligning to sequences. There's various databases you can search again. I'm just keeping all the default options. I'm not selecting anything more than that at this point. So I'm just hitting search on this sequence, and this is what I get. So I have a list of names here. I can see most of them are Cortinarius. So I think I'm pretty confident that what I have is something in the genus Cortinarius. And of course, as you know, it's a huge genus, so it doesn't really say a heck of a lot to work with Cortinarius only, but this is what I got so far. I'm getting a lot of informal names here. So the first 8 or so hits are Cortinarius per FL08. It's an informal name. It doesn't really tell me about a species. If I go further down the list, I can see, and I can also look at the statistics here. So percentage identity is a good one. We also want to look at query cover. So all of these are, most of the sequences I hit. If you get a very low coverage, obviously it doesn't really mean a lot, but 100% is darn good, and you get 100% identity, so it's very close to this. If you get into the lower levels of this list, you see some scientific names popping up at lower levels, 96% and so on. I should say that, of course, we are a public sequence database, so we are dependent on people accurately identifying sequences they submit to us. And of course, there will be errors. So when you search with a sequence in hand and you get to a list, you should never just take the first hit you get. You should look at the whole list and try and make sense out of it by looking at the context, and that lowers the amount of errors that you could possibly get. So if you want to make this list a little bit more friendly to your needs, you can hit the select column button there, and you can select specific things to show up in the column. I've just highlighted a few things here, like the description, which is this first list, the scientific name. Sometimes the description and the scientific name are not the same depending on how the sequence was submitted. So the scientific name will always be the most accurate part of that. Often the description should update with the scientific name, but for older sequences, that's not the case. We have common names, so none of these have got common names. So we do have some common names in our database. We add them not frequently, but when they're pretty well used, we tend to be biased towards American common names, although we do have international ones. I think with something like I'm Naturalist now, I have a little bit more confidence that common names are being used widely. So I tend to add more than I used to, but they're not pervasive in the database like you would see in iNaturalist. And then, of course, the accessions is always important, and then the stats about the sequence. So let's go back to our original list. So, okay, so I'm seeing my list of names here. And I'm thinking, there's lots of other ways I can look at this. I'm going into those details on taxonomy and alignments. Let's just look at the distance tree of the results. So this is a very rough and ready tree algorithm. So this is the only tree I'm going to show in this talk. So I'm sorry to disappoint those of you that wanted to see trees, but it gives you, and fast minimum evolution is a really susceptible algorithm to sequence length variations and various other things. So as always, you should take this with a grain of salt, but I think it serves a good purpose in this case to sort of survey the landscape of the things that you hit by blast. So here's my query sequence in yellow at the bottom. And it, the cluster of informal names around it agrees with what I saw on the list. And then further out, I can start to see some scientific names like Cortinarius suberythrinus over here. But some of them are, and I've collapsed some of these trees here to make it a bit simpler for display. But some of these are colored in green, and I want to draw your attention to those. Those are sequences that we annotated because they came from type specimens. And type specimens, of course, are the original specimens that were deposited when the species was named. So if you want to be confident that the name on a sequence is correct, you could have the highest confidence in something that came from the type. Of course, there are errors in those things too, so nothing is 100%, but if I see things from type, I tend to be more confident that it is correct. So in this case, I can see there is a couple of species here, but it's still fairly distant from my mystery sequence. And it agrees with what I saw when I just looked at the list. So now I can just give you something more about types here. So of course, there's this interaction between the type specimen that we get from taxonomic descriptions, and then the sequence that came from that. We have this separate database in GenBank called RefSeq that's existing from curated sequences. These are sequences that are selected because they meet certain criteria, like being from a type. And then in those sequences, we are more confident in their name and lineage. So here's sort of just a breakdown of the RefSeq ITS database. We have it for other gene loci too. And ITS, of course, is the commonly used barcode for fungi. But here, I think there's not a lot, it's a lot of details on this, but I think the number here in the bottom indicates there's more than 18,000 sequences in this database that's been selected, covering the diversity of fungal classification as much as we could get our hands on it. And this is curated by a curator at RefSeq. So these are separate sequences that's duplicated from the general number of sequences, and they selected to be more trustworthy. So here's your ITS representation of the ITS locus, the ribosomal RNA subregion with the small subunit, the large subunit, and the 5.8S, and these interspecific regions that's variable and that people use to distinguish species. When they go into RefSeq, it's the curator makes sure that the gene orientation is all the same, that the edges are trimmed correctly, and that the material is associated with a public collection or herbarium. So that's all a sign of quality of sequence and trustworthiness. So here I've just broken down all the collections in the RefSeq collection, all the herbaria and fungaria, I should say now, and culture collections that produce the data. So you can see it covers the world. These sequences were deposited from all over. I don't think there's any other message than that it's very diverse, and it's also very diverse geographically. So let's go back and do a quick search against these reference sequences only, just to make sure that we're not missing anything that could be a species. So if I run this search, I can select the ITS database and sequences from type material. Here my BLAST interface, and I can run the same BLAST again. And then I see, I confirm what I've seen before. I can see now only things that came from type material. So species, that there's also varieties here, but there's mainly species. They all start at a percentage identity of 96% and going lower. So again, this sort of confirms what I've seen before. So I don't think this is going to yield anything more than what we've produced with the regular BLAST. So let's go back to our original BLAST output. And now I'm going to start looking at the individual records. So let's look at the individual records. So I'm just taking the first one here. And now I see something interesting, right? So we've up to now only seen an informal name, but now suddenly it's got a binomial species name, Cortinarius quasi-palustris. So what's happening here? So I know about this one because I just updated this sequence last week. But what is happening here is that the taxonomy information is not propagated equally across all databases at NCBI. In this case, the sequence, the nucleotide sequence database were updated before the BLAST database was updated. So we just caught this really recently updated sequence before BLAST could actually update it. And eventually you will see it in the BLAST database. But if something like this happens, you got to decide now whether you can trust this information. So, so I also notice here in the accession, there's a 0.2, which means the sequence has been updated. So I want to see a little bit more about that. Let's go into this record. Okay, so I can go to the top of this record and just click on the revision history format. And that should tell me something. So there's various formats you can select when you get to the top of a GenBank record. Okay, and now I can see, so all the changes that's been introduced into this sequence since it started. So this sequence was produced in 2024. It was submitted to us in 2024. It's had 5 changes. The most recent one being July 29, when I made the update. So you can see the old name here, Cortinarius per FL08, and you can see the new binomial species name. And you can also see that a type has been added. So that gives me confidence that this is a recently updated record where an informal name was changed to a binomial scientific name. Okay, and if we go further down in that sequence record, you will see the holotype being annotated here. You can see the geolocation. You can see an INAT number, so it means there's some collection data on iNaturalist. And you can see it was collected by Scott Ustuni, who some of you may know. Scott sends us very often updates for sequences. So here again, let's probe a bit further. So we have the name here, and there's a link to this name. So now I'm talking about the resource I'm most familiar with, the taxonomy database. It's a database, it's actually a series of databases with names. And it only contains names, not sequences. So we provide the names for all the other records that's in GenBank. And this is the taxonomy browser. I should say this is the legacy taxonomy browser. It contains a lot of information. It's pretty dense. It's got some links to the sequences here. You can see some other information, other links to the classification. But I really want to change to the updated version of our taxonomy browser, which we're going to switch to within the next year or so. That's a bit more of a cleaner look to it. It also now includes images. So this is an image that needs to be added by a curator. We want somebody to look at the image before it gets added. It's not necessarily an expert, but we try and gather these images from as reliable a source as we can. It needs to be under the Creative Commons attribution, and we will display the license and the attribution. So if it's not a CC attribution, we will not use it. And then we have sort of a disclaimer. So if you go to a species page and you see an image that you do not like, it's possible that we could have used the wrong image in some cases. If you have a better image, please contact us and we will certainly consider updating our images. Or if there's a missing image and you want to propose an image for a species that you care about a lot, we will happily use that image. We have plenty of species pages without images, and it's fun looking through the various image databases to try and get good images for species, but it's slow moving sometimes. So here we have links to our sequences again, and I can now click on the extra information, which is a page in from the legacy browser. We have a lot of data about types here. Now it's all more separated out, so it's easier to see. And we have a common name. So in this case, this is a newly described species in 2025. You can see there Scott Ustuni described it in 2025. But together with the scientific name, he also produced a common name. So in this case, that's not something that happens very often. So I decided to add the common name under the assumption that this will become a common way to refer to the species. So I think it was useful to add the common name in this case. And then again, we can see the previous name, the informal name displayed here. And I should say that if you go to our new browser, it's very easy to download any of this information. You can download lists of species by going to fungi, to the kingdom level, and we will, through the web interface, you can get the whole list of all the fungal species, all the informal names as a single table that you can use in Excel and filter out all the stuff you don't like. I mean, it's very easy to get everything. So I need to sort of talk about NCBI taxonomy here, which is the resource I'm responsible for. As I said, it's a separate database. It's a standalone database. It only consists out of names. And we have more than 3 million names, more than 800,000 species, only for things which have sequences, but we're expanding it to things which do not have sequences yet. And we rely on external sources like Index Fungorum that somebody has shown here a page of microbank, various other worms, various other external sources with information about a species in order to populate this database. So it's really a secondary source. We rely on those primary sources. And then, of course, publications. So we have this amount of names, and then we have more than 23,000 images that we hope to extend as we go along. So that at least most of our profile things could have good images. Those images, again, should not be diagnostic, but they should give people a sense of what the species is. And then we grow at quite a reasonable clip. It's queried a lot. Most of the resources at NCBI and beyond use the names that we provide, and they need to verify when a name's been updated, like the BLAST system. The BLAST system will now propagate that name through its databases. It's like I said, it doesn't happen instantaneously, but it will happen maybe within a week, I hope, that you'll be able to see that in BLAST. And the ways that we verify these names when it's new names, it's also to rely on the rules by the codes of nomenclature, these rules that's determining how you describe a new taxa, new species, and other taxa. So we have four codes that we have to deal with. There are other codes as well, but these are the main codes for prokaryotes, algae, fungi, and plants, animals, and then viruses, which has seen a lot of changes recently. So don't get me going on viruses. All the species names for viruses has recently changed, so it's almost all. It's a big deal. So here we have taxonomy, bird's eye view of our taxonomy. So this is an external site life map, but I think visually they've made a very good job of being able to traffic or to go through the classification that we provide. But this is showing you all the organismal nodes that we have in our database, not viruses. But here you can see in context, here's fungi, in context of all the other names that's in the database right now. So we like to believe that we are at the center of this web of all these interconnected databases at NCBI, because taxonomy touches everything. And once you have the name for something, you can actually learn something about it. So NCBI, I'm zooming out a little bit now, out of course now as well, as many resources. We are very much focused on human health, but humans do not live by themselves. They are part of an ecosystem. All of these other parts of life have data associated with them, and we, as the taxonomy database, are interested in making the names for those as accurately as possible, making the data that people submit to us as accessible as possible. So, of course, for all this data, you need many resources. And this is just a few resources that were recently updated over the last year. We have a blog called NCBI Insights, which regularly provides updates on new things that are being done at NCBI. So here, BLAST, of course, BLAST has many updates. The publication database, PubMed Central, which has got full text journals, but also PubMed has got abstracts, and there are some fungal information in there as well. And then we did a lot of fungal taxonomy and well, yeasts and bacterial and viral updates this year, so people noticed that. And then every year you have the more medically orientated databases like clinical trials provides a lot of information about clinical trials. There's a database, a dbGaP. It's a database for genotypes and phenotypes and various other resources. So if you're interested in seeing how things are updated, check out this blog. And of course, I should mention that as we build new things, priorities shift, and we have to retire something. So this is a recent resource that was retired in January. So you might see that some things that were there before, because of usage patterns and priorities, change. So another important point is that the data that we have in GenBank does not all come to us. We are part of an international partnership with, and that's probably one of the longest standing scientific partnerships. It's a really good model for how to collaborate internationally. But we have data from Japan and Europe that we mirror. I think GenBank takes in about 80% of everything, but we mirror all that information and they mirror ours, and there's daily exchange of data. Each of the three partners in this case have got their own submission tools. And so the submissions might be slightly different according to the priorities of each partner, but the format of the data will look exactly the same when it's being mirrored. So you would not necessarily notice something when you look at a sequence record that was deposited in Japan versus one that came to us. But the submission experience might be different, and you might they might prioritize different things. So here we are at submission. So this is our submission tool. We have separate, and this is a really new interface. They've really spent a lot of time in trying to improve the usability of this interface and make it easier, because it's a common complaint that people are finding the submission part of the process difficult, or it's too slow, or it's hard to understand. So a lot of effort is going into this part of the process. So here, one way to make this more user-friendly is to separate submissions out into different groups. So if you've got a fungal sequence, of course, you would choose the eukaryote, and then you would get particular questions. So you would get a question about if you're using ITS. It, of course, will not apply to prokaryotes or viruses. So you would get specific questions about ITS. So here's an example of a question that you would get if you submit. More details are, you could use part of the ITS, the full ITS. There's other information on other barcodes here, like COX-1, that's used in animals, or chloroplast information that's used in plants. There's lots of ways to FAQs for questions. There's a whole knowledge base. And then if you've exhausted all those possibilities, you can proceed with your submission. So here's one explanation of what's needed for an ITS submission. It's, of course, eukaryotic, but I want to highlight two important pieces of metadata. That's the collection date and the location. I think previously, up until the last 2 years, we're making this information together with our international partners. We're making this required information. Previously, you could ignore this and we would accept your information. But now you need to tell us why you cannot provide that due to privacy or security concerns, but you need to provide a reason. And there's a set of reasons that we will accept. But we hope that this will provide more complete data. And as you produce, maybe you're collecting something and somebody else is doing the sequencing for you, you still are involved in that process and in providing that information for the location. And in order to continue to make sense out of that sequence, the date and the location is essential, besides the name. I think it's one of the most important things that one could do to make the data reusable. So this is part of what's called the FAIR principles. It's been around a few years now. It's an attempt to have data being more accessible to people and have databases ascribed to these principles. So if you're not familiar with it, it stands for findable, accessible, interoperable, and reusable. So of course, we try and follow all these principles. I'm focusing for this talk on the reusability part by mentioning the ways that submission plays an important role in that. And as a database, we need to then make the data findable for people that come afterwards. And I think the other part of this process is that when the story of that sequence, and here's just one entry for when this story of our mystery sequence is told, we know that it's got a name, it's got a specimen to it that came from a type, and it's got now a new RefSeq entry that was just made for it today. So it will go into the selected RefSeq database. If you go and search on iNaturalist, you can see Scott's samples already. There's really 5 collections on there with pictures starting in 2023. So it didn't have a name back then, but the name's been updated, so that's great. There is specimens in the MycoPortal database submitted to herbaria in Florida and Tennessee. And then there's an image, several images, but this is one. So St. John's Webcap has just been born as a name. It's always been there, but I think there's a good chance that others can come and add to the story of the species, or say it's not a species and change it to something else. But I think the important part is here that we have data that allows others to come and reuse and build upon this. And I hope all of you that are involved in this process will be inspired to do something similar. So with that, I will end with acknowledgements. I'm just showing the members of my team. We have several curators covering different parts of the taxonomy, plants, invertebrates, bacteria, viruses, all people behind the scenes that are very motivated to make the names as correct and as accessible as possible. I've talked about the GenBank team's work and the RefSeq team's work here as well. All very dedicated people, other NCBI colleagues. And then I should highlight the work of the NCBI Help Desk. If you have a concern or something that you do not understand, they're really good at answering questions or passing it along to one of the other experts. So if there's a question about some of our taxonomy or an image that's incorrect, write to the help desk and I will see it and I will respond to it. And I'll be happy to answer any questions. I think I'll stop my screen share now. Hopefully everybody's still there. I didn't talk to myself all this time.
No, we're still here. Thank you so much, Conrad. My word. Again, like I said, after Emily's presentation, I'm so glad that we record these presentations. I'm going to have to go back and check that out. Watch this over another time or two to let things sink in. I am going to change the security settings. If anybody has any questions, I'm going to change it so you can unmute yourselves.
So for instance, I did check, and we do have sequences for Trichoglossum octopartitum. So if you happen to sequence that sample, you could verify and see if it matches those sequences, or if it's maybe a different Trichoglossum.
Tom, I had a comment and a question. One, Conrad, I'm super excited for the taxonomy browser update. I think it's great. Do you have an idea when that's going to come out?
It's already out.
It's already out.
It's just not obviously linked to it right now, but if you search on data sets, you'll see a taxonomy, this taxonomy link. Just search on NCBI data sets and it will come up. If you go to the records themselves, it's still linking to the old browser, but we're in the process of changing that over the next 6 months.
Okay, yeah, no, that's fantastic with the images.
I'll be happy to provide you with links. I mean, there's also command line tools for those people that want to get everything by command line. But all of you, if you have images that you want to share, that you think is really a good image for a particular species, I would really appreciate having those images. And of course, we do show it with the license and the attribution on those pages.
Yeah, that's great.
So yeah.
So more and more mushroom groups and mushroom clubs are beginning to extract DNA and do Sanger sequencing, especially the ITS region of mushrooms that they're dealing with. When they are putting these sequences into GenBank, first of all, do you encourage them to put the sequences in GenBank? And second, do you have an opinion on how they should format names? Should they use their best guess for a species or just say species or?
If you're not sure about the name, use the lowest level that you're comfortable with. So go to genus or family, and then we'll just put it in as a spin name. Like you could see that Cortinarius that came in, it wasn't identified as a species at the beginning, but it came in with it actually had a unique string. It's the Ohio Mushroom Lab's data. Steve Russell, I think, is also a lot of his data comes through there. So they've used a unique string in that case, but I think it's important that you have voucher information that's at least an isolate, so we can actually determine after the fact if we want to update those names. So if you're not sure about an identification, rather go up to genus, I would say. And then also the quality of the sequence, please trim the edges. I think there's a lot of really crappy things on the edges and people want to get as much sequence out sometimes. When it hits it affects the way that the BLAST algorithm sees things. So rather trim the crappy parts out and leave the good sequence in.
That's a good
I
suggestion.
would definitely encourage people to submit, and then also the location information is crucial. It's really the collection date and the location information. That's really the value there.
That's great. And I guess, I mean, I know the answer to this, but well, part of what I wanted you to come on, why I was excited that you were coming to talk to us, is that a resource like GenBank or these government resources are usually faceless and nameless, and they're just a database. And you talk about going to GenBank and doing a blast search, but there are people behind all of that.
Yes.
And so I think it's important as folks are beginning to use these resources more, that they know that there are people there that are actually doing the work behind the scenes.
Yeah, and I think that's why I highlighted the help. I wanted to show the picture of the people that's doing the work, but also the help desk. People sometimes think they're not going to get an answer, and they're always surprised when they do. I think a help desk is really good at getting people answers about certain things. So there's a lot of information if you really want to go into webinars and all sorts of things. And we do work for experts, but we do it for everybody, really. It's we have teachers and students coming to us, so we need to be able to handle everybody and provide information for everybody.
Great. Well, thank you so much. You might be seeing some sequences from us.
I do. I do see a lot of ITS going in. I mean, it's probably one of the biggest single streams of submission now are mushroom, collecting groups, sequencing things and putting them in. So yeah, if you have questions, particularly about submission, just let me know, and I can see what I can help. I'm not working for submission, but at least I can go ask them if I don't have the answer.
Yes. Well, thank you again.
Yeah, I enjoyed this. Thanks for inviting me.
Thank you very much.
Okay, everybody, we are just about on time. Look at that. I thought we were going to go way over tonight, but here we are at the 9 o'clock hour. Does anybody have any parting questions before I close the meeting? Okay, thank you so much. I will be seeing some of you tomorrow night at our board meeting, and for the rest of you, be seeing you soon, hopefully at Sequenota. All right, have a good evening. Goodbye, everybody.
Bye.
Goodbye, Team Fungi.
Bye.
Good night. Bye.